You're listening to the Assembly on Allergy, Immunology, and Inflammation podcast, brought to you by the American Thoracic Society.
So hi everyone, welcome to the American Thoracic Society podcast. Our topic for today is on resolution of inflammation in acute respiratory distress syndrome, also known as ARDS. My name is Yong, an assistant professor at National University of Singapore, along with my co-host, Dr. Filiz Korkmaz, microbiology endowed scholar and assistant professor at the University of Iowa. We are very fortunate to have with us today three esteemed panelists. They are Dr. Bruce Levy, Hersey Professor of the Theory and Practice of Physic at the Harvard Medical School. He's also the Executive Vice Chair of the Mass General Brigham. We have also with us Dr. Kimberly Gowdy, Associate Professor at Ohio State University and the Associate Director of the Medical Scientist Training Program. And lastly, we have Dr. Benjamin Singer. He is the Lawrence Hicks Professor of Pulmonary Medicine at the Northwestern University. He's also the Vice Chair of Research at the Feinberg School of Medicine. So I'm going to pass to my co-host, Dr. Filiz.
Hi, thank you everyone for joining us today and for the wonderful panelists we have for this really important topic, I think, in the context of understanding acute respiratory distress syndrome. And not only, the primary inflammatory mediators, but also the factors that go into, resolving that inflammation once it occurs. And I'd like to just kind of open it up to our panelists, kind of go down the line and see, what would you say, I guess, to define ARDS first? What would you say is the definition and primary causes? We start with Kim.
Oh,
You're my
yeah,
first
I mean, so thank you all for having me on this panel. I will say that I think of ARDS, mind you, more from a mechanistic perspective in my research program. And in that aspect, a lot of it is driven by kind of unchecked inflammation. And then that unchecked inflammation in my hands, results in basically a physiological dysfunction of the lung.
Okay, we can hand it over to Bruce.
Sure. Thank you again, Filiz and Yong, for involving me to be part of this panel. It's an honor to be here with Kim and with Ben. And it's fun to think about resolution of inflammation as we think about ARDS. Of course, there's a clinical definition of the modified Berlin criteria for ARDS. So it's very clinically derived. But I think today we're here to talk more clinical translation and mechanism, and with a focus on resolution. And so in that context, as Kim was describing, I think of ARDS as inflammation gone wild in the lung, and to the extent that what should be a protective mechanism for our bodies is we're now drowning in inflammation to the point where we can't get enough oxygen. So the braking signals, the counter-regulatory signals are not functioning properly. And so from that perspective, after thinking about it as a clinical entity and the modified Berlin criteria, from a mechanistic perspective, especially the early events in ARDS, it feels quite maladaptive, as what should be a protective function of inflammation is now filling our air spaces to the point we can't get oxygen anymore.
It's both easier and harder to go last after the comments from Kim and Bruce, with which I completely agree. Something that I remind the trainees that I round with in the ICU and people who join my lab is that ARDS is a syndrome, not a disease. It's something that we recognize by a collection of clinical criteria that we can model experimentally in model organisms, but that ultimately the clinical entity is still really poorly understood. It's a syndromic definition. It's not a disease definition. And when we start talking about some of the things that I think we'll get into in this podcast, and this is another thing that I talk about with people who are joining the lab, is a lot of words that begin with R right? So we talk about recovery, right? So that I think about that as the physiologic restoration of homeostasis as a patient recovers in the ICU and can be liberated from life support and can be continued down their care pathway, or we see recovery of physiological parameters in a model organism, like in a mouse model of ARDS, or experimental acute lung injury. And then what we're going to talk about today is resolution. So not just reversal of those initiating inflammatory signals, but actually the active processes that drive the clearance of inflammatory cells and the restoration of normal inflammatory function or functional inflammatory function that might work in pathogen defense, but not drive the collateral damage of ARDS. And then there are these other R words like repair, which is linked with resolution. But when I think about repair or regeneration, I think about rebuilding, another R word, the damaged tissue. And that involves cellular processes of differentiation, proliferation, migration. And again, these things are linked, but since they all start with R, I think it's a good framework to start our discussion.
I
Maybe
completely agree.
I'm going to expand on that, Ben, a little bit, and let's add a few more R words. But the resolution of inflammation, as Ben is really driving at, is an active process with specific cellular and molecular and even organ level mechanisms to convey this natural process to respond to, a challenge, if you will, with a focus now today on the lung. But so we think about inflammation as having pillars of response that have been defined since the first century by Galen Celsius, and picked up on by Galen. And but so for resolution, the R words that Ben was beginning to kind of list, we think about removal of microbes, dead cells, debris, restoration of vascular integrity and perfusion. Both of those are really relevant in ARDS. Regeneration of tissue, which does happen in the lung in this context, and certainly things like remission of fever, relief of pain. So just wanted to add a few more R words to the alphabet that Ben is beginning to
Love
build
it.
for.
Excellent. I completely agree. So I guess, in the context of going more into some of these cellular mediators and mechanisms of both pro-inflammatory and, factors that are promoting ARDS versus those that are actively resolving ARDS. I guess I'll start with Dr. Gowdy in that. What do you think are the primary, pro-inflammatory mediators that you often see that are the sort of the factors that promote acute respiratory distress syndrome?
Yeah, so I would say one of the things we focus on is kind of the polyunsaturated fatty acids. So these are, either your N6 polyunsaturated fatty acids, such as arachidonic acid, linoleic acid, etc. And so, those are kind of through enzymatic processes or even the production of reactive oxygen species made into oxylipins, such as your prostaglandins, leukotrienes. We have tons of biologics on the market that target these, as well as just traditional therapeutics. And so those really, I feel like, are some of the first actions that happen to kind of drive inflammation. And so, they call in, obviously, your neutrophils, your monocytes. They can even trigger the inflammatory response of a lot of the resident cells of the lung, too. So I feel like they're kind of the first actors. But one of the things, at least we've seen in some of the ARDS research we've done, is then they become unchecked. And, in order to start the resolution process, there's this process called lipid class switching. And that's when the body stops using these N6 PUFAs as fuel and starts using kind of some of more of the N3 PUFAs as fuel. And that actually can initiate the resolution process. So I would say, in my mind, kind of the drivers of some of the inflammation in my mind starts with kind of this PUFA metabolism that can really kind of not be turned off.
And you touch upon one of my favorite cell types is the neutrophil. And I know, often they're associated with this sort of pro-inflammatory processes. And I'll open it up to both, to Dr. Gowdy, but also to Drs. Levy and Singer, in that, do you think that there is some role for them in the opposite, in sort of the pro-resolving part of this process, given the fact that we have such insights into their sort of their heterogeneity these days?
Yeah, so I'll start. I would say that yes, they're essential because without them, you don't get resolution initiated. So it really is very important for them to be produced, to be, in the local environment to really kind of drive that return to tissue homeostasis. So, and I will say that I think of also all of it like a seesaw, because we have the host events and then we have the inflammatory response. And so you need that seesaw to kind of find equilibrium to really kind of get that tissue regeneration to start.
Yeah, I agree. I've become really interested in neutrophils over the last couple of years. I think they're clearly the most abundant yet most poorly understood immune cell that we see in inflammatory lung injury, right? The hallmark of the alveolar inflammation in severe pneumonia, of course, and ARDS more broadly, is a neutrophilic alveolitis, but we tend not to focus on them. And I think the heterogeneity of neutrophils that was just mentioned, I think that's a huge point. These are not just pre-programmed robots that show up and explode and cause damage and then need to be removed. I mean, there's so much more to the biology. And I think there are some really interesting emerging data about both systemic and local signals. So signals within the alveolar niche of the inflamed lung that control both neutrophil fate, so longevity. They probably live a lot longer in the tissue than they do in the circulation, and also their function. So moving more from kind of classic mediator degranulation mediated effects toward other effects that, like was mentioned, can drive resolution and initiate repair processes. So yeah, huge amount of work to be done in neutrophil biology.
So I have a few comments here. I mean, as we talk about the kind of the initiating events, Filiz, that you were asking about, we learned from COVID, this terminology of cytokine storm, and it's kind of an unregulated just burst of pro-inflammatory peptide mediators. There's when the lipidomics was done during COVID, we found that it was the same. There was a lipid mediator storm with all of the factors that Kim was listing, but as she was saying, like in an unregulated manner. And so just tremendous confusion in the lung of how to respond because it's being overwhelmed with all of these pro-inflammatory mediators, and then leakage permeability, cell damage, oxidants, etc. So neutrophils, so I have to be careful how I phrase this because I have some private information that's about to be published on this. But there's evidence that will become available shortly on a pro-resolving subset of neutrophils that in the lung emerge during resolution. And in model systems, they're not really found in the circulation. So as Ben was saying, in the alveolar niche, they're responding to tissue level signals that inform a change in the transcriptome of the neutrophil, the products it's generating, how it interacts with its neighboring cells, etc. So very soon there'll be public information about this subset. We're very excited about it, but I think the fundamental point here is, as everyone has mentioned, the neutrophils are really fascinating. Number one. Number two, that they are not just robots, that they do change in response to environmental cues, and they can participate not only in, importantly, host defense by an activation sequence that can lead to kind of innocent bystander tissue injury, but also, importantly, they can also participate in the resolution of an inflammatory response. And, there's some kind of clinical sense of this because, neutropenic patients can actually also get ARDS, and they don't do as well. They don't, they have longer recovery times. They have more severe, ARDS, generally speaking. So I think there's a clinical correlate here that helps us understand this. The last thing I wanted to say on this point is there was a fundamental observation in the 1800s by Elie Metchnikoff that macrophages, he identified phagocytes, of course, the father of innate immunity, that they could engulf dead cells or dying cells. And he didn't kind of further elaborate on that, but these observations were picked up in the 80s importantly, by John Saville and Chris Hazlitt and Peter Henson, when they were working in Edinburgh, and defined really efferocytosis. And it's really that cellular process, and they defined it in pneumonia. So in ARDS, this definitely happens as well. But this fundamental cellular process is like the match that ignites really the resolution paradigm in healthy responses. So I think it's just important to put that in context, that macrophage-mediated clearance of apoptotic or dying cells is an important early event that's triggering, the resolution processes.
Yeah. Go ahead.
Okay, yeah, so that's interesting. So, we have the pro-inflammatory responses, but we have mentioned a few times now about pro-resolution, pro-resolving. So just, for the audiences who don't really know what they are, I figured that I could ask Dr. Gowdy to start first, if you can share more of these N3, PUFAs that you mentioned earlier about the pro-resolving molecules and, what they are and what they can do.
Yeah, so sure. Yeah, so the specialized pro-resolving mediators are basically a class of oxylipins or lipid mediators that are very important in driving in the resolution of inflammation. A lot of them do promote efferocytosis, like Bruce was mentioning, and they also, block NF-kappa B. They can, upregulate the production of anti-inflammatory cytokines such as IL-10 and even antioxidant responses too. So there's the D series, which are, derived from DHA, which is again an N3 PUFA. So that's, your resolvin Ds, merisins, protectins. But then there's the resolvin Es, or the E series, the resolvin Es. And then there's even, some that are produced from some N6 PUFAs, such as the lipoxins. So I say N3 PUFAs as driving kind of these SPMs as like a general category, but do know that some of them are actually from the N6 lipid metabolism. So, and how a lot of these specialized pro-resolving mediators do their action is they actually bind to G protein coupled receptors on cells and kind of initiate this downstream signaling that results in kind of the downregulation of inflammation or the efferocytic response of phagocyte.
That's interesting. So on that notion, before we get to macrophages that Bruce mentioned earlier, it's that I think some papers are starting to suggest that this anti-inflammatory cytokine called IL-10, interleukin 10, right? I wonder, Dr. Singer, if you have any, comments on, do you think they're actually pro-resolution?
Yeah, I think, you can get a lot of information from single gene knockout models in mice, right, where you knock out IL-10 in specific populations, and you can change over the course of an injury model different recovery phenotypes, right? So certainly there's a role there. What it's doing is more complicated. So so in my lab we talk about regulatory T cells, which is a cell type we can begin to talk more about, but it's a subset of CD4 cells that has a counter-regulatory role against inflammation. It's kind of the traffic cop of the immune system. So it regulates the flow of inflammation in an adaptive way, ideally. And one of the things that regulatory T cells make is IL-10, and there's a mechanism involving IL-10 signaling through into macrophages, which engages a RAC1-dependent mechanism to drive efferocytosis. So that's a way to clear neutrophils that's Treg dependent and involves IL-10. So that's just one kind of straight, narrow pathway, but it's a pleiotropic cytokine.
Interesting.
I might mention that it was also the subject of the Nobel Prize in Medicine this year.
Yes,
That's correct.
So,
That's correct. So
is working on very important research.
Yes.
yeah, it was a fun day in the lab when the Nobel was announced a few weeks ago. So that's right. It was awarded for the discovery of mostly the immune regulatory functions or the immune suppressive functions of regulatory T cells this year.
So, Dr. Levy, earlier you mentioned macrophages, being involved in efferocytosis, and I guess the notion more of efferocytosis is this state of the macrophages where they become more like M2-like kind of macrophages. So, what's the role of these M2 macrophages in resolution?
So macrophages are pretty interesting cells because they respond briskly to environmental cues, and whether that's cytokines that drive a transcriptional program or it's dead cells that activate macrophages in different ways. Their recruitment signals are interesting, and they are they have a pretty active transcriptome, so they can change very rapidly, and it's there are paradigms that have been established that call like an M1 macrophage or an M2 macrophage based on defined transcriptional programs. But honestly, it's a little more like a kaleidoscope rather than a fixed determinant of one versus the other. M2 macrophages are a little easier to define in preclinical, like mouse models of lung injury or infection, a little less straightforward in humans, but they have a counter-regulatory function that includes efferocytosis, includes anti-inflammatory mediators. It includes, inducing the expression of dummy receptors to soak up cytokines that are laying in tissues, et cetera, et cetera. So there's several functions of these cells that, they're glorified janitors in a way to help clear out the debris and the mess that's been left behind by the battle of inflammation that's raged. But they have many functions. M2s are kind of a classically defined macrophage phenotype that is a counter-regulatory macrophage. So we've talked about neutrophils. We've talked about regulatory T cells and macrophages. There are other cells, so of course, that can participate in resolution. And in many ways, pretty much, if we just focus on the lung, basically structural cells of the lung can participate. Natural killer cells can participate. Even like sensory neurons can be part of this. And innate lymphoid cells can participate through the production of things like amphoregulin or other kind of mucosal restorative growth factors. So, if you think about the cytokines, there's more than 35 different cytokines that have been defined, and there's chemokines, which are another huge array of kind of pro-inflammatory peptide mediators. We're still learning about the complicated array of pro-resolving mediators, many of which Kim listed and Ben referred to, and the cellular mechanisms, which of course in health have to be as robust to stem the tide of inflammation. Otherwise, we'd all end up with chronic, inflammation that doesn't resolve. So now those counter-regulatory functions can be disrupted by environmental or medications or other factors, and then they can be disabled in the setting of disease, with aging, etc. So there's unfortunately it's a fragile system that can be overwhelmed. But so the macrophages, circling back to this, are very important and central to resolution. But there are many cells that participate in this, cell types that participate in this response.
I'll add a few comments about macrophages. So the M1, M2 paradigm, I think, is really helpful for in vitro studies, right, where you have a defined stimulus and then you can define a transcriptional signature, and maybe that overlaps with some subpopulation you see in vivo, but it really at its heart is a reductionist in vitro signature. A really helpful way for me to understand macrophage biology in the lung is to think about their ontogeny. So you have a population of tissue-resident alveolar macrophages that derive from embryonal tissues and seed the lung and replenish over the lifespan. And then in the setting of injury and inflammation, there are cues released from the lung that recruit blood monocytes to the lung, which then enter the lung and rapidly become alveolar macrophages. And these monocyte-derived alveolar macrophages in the context of injury are highly inflammatory and profibrotic. But over time, through the interaction of many of the signals and cells that Bruce was mentioning, they will begin to adopt a transcriptional signature and an identity that more resembles a tissue-resident alveolar macrophage. So we're all kind of alveolar chimeras of those different populations, but ultimately it's the microenvironment that determines the long-term identity and plasticity of those cells.
If I could add to that as well, I think that, also if you look at the process of efferocytosis, so when a macrophage eats a dead cell, that kind of like, I think of it like a lipid package that it takes up actually induces arginine metabolism, which induces actually some of the hallmarks of what folks, I guess, do refer to as an M2 macrophage. So I think that's how they've got kind of lumped together. But I do think, again, like that, metabolism, you are what you eat, right? So I feel like that actually plays into what they produce after they either clean up an environment, whether it be from a pathogen or from an injury and needing to, return tissue to homeostasis.
So one more question on my part before I pass back to Filiz. So I think we mentioned so far like the pro-inflammation phase as well as the pro-resolution. So I think one common question, that usually we have is, are these phases of pro-inflammation and pro-resolution, do they occur separately? Are they occurring in parallel? So perhaps I open to all three, panelists to comment on that.
I'll start. I was gonna say, I think that they're always kind of in cooperation. So again, I think this comes back to the balance. A lot of what we've seen in the lung of mouse samples as well as in human tissues is that, kind of there's some that go up and there's some that go down, but I don't think like one is completely, dominating and then the other turns on. I think it's just, who's kind of who's on first in my mind. But yeah, I do think that they're constantly cooperating, and there's a lot more I think we need to understand, especially with the lipid mediators, of how they kind of cooperate and how one kind of dominates a certain phase of the initiation of inflammation or the resolution of inflammation.
I'm happy to weigh in. This question really dates back to some of our earliest work that actually uncovered the lipid mediator class switching that Kim referred to. In the work of Derek Gilroy and others, they found early on a pivotal role for cyclooxygenase 2 in kind of this conversion, if you will, from pro-inflammatory prostanoids to counter-regulatory lipoxins at the time. And we began to look at this at the same time and identified that the early events of inflammation actually importantly lay the groundwork for the development of pro-resolving biochemical pathways that enable the production of those, SPMs, specialized pro-resolving mediators, and then the cascade of downstream cellular events. And it turned out that PGE2 was a very important early mediator in this regard, and PGD2, prostaglandin D2, could also serve this function. And so they work through cyclic AMP response elements to induce the production of an enzyme called 15-lipoxygenase, which is important for a cassette of these SPMs and their production. And if you inhibit early COX-2 activation and early prostaglandin production by the COX-2 mediated pathway, you can totally disrupt this process. And so we entered the scene as it related to lung injury by the development of a survival model in which we unilaterally injured the lung with hydrochloric acid, modeling a gastric acid aspiration event, but only injuring the left lung by selective installation of the acid into the left mainstem bronchus in a technique that was taught to us by Jay Misgard, who was a pneumonia researcher. And in that setting, the animal was not injured on the right side, so they could survive. And we could now not only look at right-left comparisons, but look at the counter-regulatory and pro-resolving time course. And when we inhibited COX-2, either pharmacologically or genetically with COX-2 defective or deficient animals, what we found is that if you stopped your experiment early, as most of the lung injury work at the time had done, because mice don't survive really beyond about 6 hours, 8 hours because of hypothermia in the early kind of lung injury modeling work, then you would look at that and you'd say, wow, this is great. It's inhibiting inflammation, and we should think about this as a therapeutic. And in fact, at least 5 different clinical trials were born out of that observation, all of which unfortunately failed in ARDS clinical research, human clinical research. But if you were to keep going and allow the animal to go through its natural resolution arc, what happened is that you markedly delayed resolution because you uncoupled the early events of inflammation from the later events of resolution. And so John Saville and Charlie Serhan came up with this moniker that alpha programs omega, thinking about the early arachidonic acid related pro-inflammatory events, importantly laying down the biochemical framework for later omega-3 fatty acid derived specialized pro-resolving mediator formation. So there is definitely this kind of balance or in homeostasis where you've got kind of a back and forth that Kim referred to. But in disease settings and in model systems, you can disarticulate the two, and it becomes very clear that importantly, in a regulated response, early events in inflammation are actually important for healthy and timely resolution.
And one thing that Bruce had mentioned earlier in terms of host factors that predispose patients to ARDS is age. So that's sort of the next topic that I kind of wanted to cover, and maybe we can start with Ben on this one. So what do we know these days on how these factors may be changing with age, such that patients are more predisposed to ARDS?
Yeah, age is enormous, right? I mean, it is far and away the biggest risk factor for pretty much every common lung disease other than inherited ones, right? If you look across fibrotic diseases, emphysematous disorders, and acute disorders, right, age is the biggest marker for susceptibility to both getting a severe disease and having poor outcomes from it. One way that we've started to think about this in our lab is thinking about the hallmarks of aging, which are mechanisms that are associated with chronological age that may impart susceptibility to biological changes in age over time. Is to think about events that occur that are cell autonomous, in other words, that occur within cells as a consequence of them just existing over the life course, or as a cell population over the life course, versus cell non-autonomous changes, which are imparted through environmental interactions and the like. And so, like I said, we're interested in regulatory T cells as a key necessary factor in driving resolution and repair, kind of serving not just as the traffic cop function that I mentioned, but also as kind of an orchestra conductor. There aren't many of them, but they stand up at the front of the orchestra and try to get all of the different instruments playing in harmony again, with the analogy being restoration of homeostasis in the lung. And so we had a question a few years ago about whether the age-related alterations in the regulatory T cells that participate in resolution and repair are cell autonomous or not. And so what Dr. Luisa Morales-Nebreda did when she was a fellow in our lab was to do some really elegant isochronic or age-matched and heterochronic age-mismatched adoptive transfer experiments, taking young cells, putting them into old mice and vice versa, and then doing all the comparisons. And what she found was that there is a cell autonomous change in regulatory T cells. You can take a young regulatory T cell, put it into an old mouse, and it will promote recovery, right? It will retain the memory of the age of the mouse from which it came. Similarly, you can take a regulatory T cell from an old mouse and put it into a young mouse, and it will actually worsen the outcomes of that mouse. In this case, it was in the influenza pneumonia model, so it retained not just a lack of function that it acquired with age, but probably a deleterious function that it took with age. And that probably has to do with stability of the lineage and some of the epigenetic factors that we've identified that determine Treg biology. So that's not the entire story about aging and lung injury, but I think it's at least a paradigm to start thinking about how the hallmarks of aging, in this case, like epigenetics, change lineage function of key cell types in cell autonomous or cell non-autonomous ways.
And just to build on that, I will say that I think the lipid metabolism and I don't know specifically if it's been studied in the lung, but I know the lipid metabolism of, peripheral blood cells, et cetera, has been studied in kind of young versus old people. And one of the top genes that's actually methylated with age is of the elongation. It's ELOV2, which basically allows cells to take up some of those N3 PUFAs and incorporate it into the cell membrane. So basically providing fuel for those enzymes. So I do think that maybe the loading of the cells with these different PUFAs, changes with age, and then that can actually change the resolution responses too.
Yeah, in experiment, similarly, Kim, experiments done in Charlie Sirhan's lab have demonstrated that older individuals underproduce SPMs. So Charlie Sirhan, just for background, really identified and structurally elucidated each of the molecules that Kim referred to up front in this genus of endogenous specialized pro-resolving mediators, and has done a lot of the important work of linking them to the framework of resolution biology that, again, has existed for a while. But in terms of aging, in work with Mario Romano, they looked at older individuals and found that they underproduced relative to younger individuals, SPMs. So I think carrying through from all three of us, from the kind of cell autonomous observations that Ben's and his group have made, and Kim's important contributions around the methylation events in fatty acid metabolism in the cell, to actually the actual products, we see that aging has a marked impact on this homeostatic system.
All right, so I think that we could all probably talk about this all day, but for the sake of time, I would love to get all three of your thoughts on basically the biggest gaps in knowledge we have right now in the context of resolution during ARDS, and how might we incorporate that into developing new therapeutics? Because I think that's a big avenue these days of where we sort of lack is that some of these pro-resolving factors, because we do have a lot of those sort of anti-inflammatory biologics at the moment. We can start with Kim.
I'll start. Yeah. Yeah. So I will say that one thing that I think is kind of the future is the heterogeneity of ARDS and how that might change kind of the resolution of inflammation with ARDS. Because we kind of group them again as a syndrome, and we have clinical criteria to meet this syndrome, but whether it's a bacterial pneumonia, a viral pneumonia, acid aspiration, sepsis, trauma, etc., I think that that's going to have a very different resolution paradigm that might lead to different mechanisms of resolution and also different time courses too.
All right,
Ben,
Ben?
do you want to go?
Yeah, this is another thing we could talk about all day. I think one of the interesting paradigms that I think still has to be worked out is, and I'm going to oversimplify it, is whether you need to resolve inflammation to allow repair to occur, or whether you need to repair to allow resolution to occur. And they probably co-occur, right? That's the simplest idea. But the idea that you could use a pure anti-inflammatory drug or modulator without driving some amount of lung repair probably explains part of the reason why we haven't seen such benefit from anti-inflammatory therapies broadly in conditions of lung injury. You have to rebuild the lung, you have to resecure the epithelial barrier at least to be limiting some of the signals that recruit inflammatory cells like monocyte-derived alveolar macrophages and inflammatory neutrophils and begin those resolution processes. Similarly, repair probably cannot proceed effectively until the inflammation has become at least a bit more quiescent than it had at its peak.
I completely agree. I think Ben laid it out, and Kim laid it out really well. Big gaps here. Bedside diagnostics, precision diagnostics. Where is, how can we determine where somebody is in their ARDS inflammation resolution journey? We don't have that really. And then I puzzle around whether molecular therapy or even cell-based therapy would be a way to go here because of all the events that Ben was starting to lay down. I mean, we've got to impact leukocyte recruitment, leukocyte activation. We've got to impact barrier integrity. We've got to impact actual clearance of all the debris and the fluid. We have to encourage laying down transient matrix as a scaffold for repair of the lung. It's hard to think of one drug, one chemical that would do that. Now, there are cellular orchestrators, and we've talked about some of them. Ben has mentioned regulatory T cells, but it does, in my mind, raise a question of whether, therapy is going to be multimodal down the road, either with multiple different, molecular agents, or whether it'll be cell-based therapies as we intervene here. But it's a really complicated pathobiologic process. It's hard to think of one molecule that would address all of this and speed the resolution, with the potential exception of prevention or very early intervention. So somebody who is having a surgery and is at risk of developing ARDS from aspiration or what have you, getting something up front that might mitigate the severity or prevent the development of ARDS. But once it's fully blown, it's a challenge. And it remains a challenge right now. And there's no clear path, I think, to definable therapy yet.
All right. Well, thank you all again for all your valuable insights on this really, really important topic. I think, there's a lot of directions to go in the future, in terms of heterogeneity, age, different cellular mediators, kind of what regulates that switch between pro-inflammatory versus anti-inflammatory versus pro-repair. And I'm really looking forward to all your insights in the future. So thank you again for joining us today for this podcast.
Thank you for joining us today. To learn more, visit our website at thoracic.org. Find more podcasts from the ATS assemblies on YouTube. Don't forget to like, comment, and subscribe so you never miss a show.